A-Level Biology 9700, Paper 4, Variant 1, October, November 2024. Question number 9. Question number 9 is about lichens. So, lichens are found growing on trees, walls, rocks, and soil. Lichens. What are lichens? Lichen is a hybrid colony of algae or cyanobacteria. Cyanobacteria which perform photosynthesis. So they're living symbiotically among filaments of multiple fungi species along with yeast and bacteria embedded in the cortex or skin in mutualistic relationship. Mutual relationship or mutualistic relationship or symbiotic relationship. These are the mutual relationship in which both host and guest relationship in which both give benefits to each other. So here question is figure 9.1 shows a lichen of the genus Usnea. Usnea can tolerate only low concentrations of sulfur dioxide and does not grow in places where the air is polluted with sulfur dioxide. Air is polluted with sulfur dioxide, it means sulfur dioxide causes pollution in the air, and when this sulfur dioxide dissolves in rainwater, it is changed into sulfuric acid or sulfuric acid. And when this water or the rainwater with dissolved sulfuric acid or sulfuric acid into that water, it fall on the ground. It is cause of acid rain. So Usnea is composed of a mixture of two types of cell. So it composed of two types of cell. They are photosynthetic cells. They are classified into Protoctista. Fungi cells that are classified into the kingdom fungi. Fungi. People pronounce it differently, fungi or fungi. It depends on you. So outline the characteristic features of the kingdoms Protoctista and fungi. So the kingdom Protoctista are eukaryotic. So very simple question. We have to only outline their characteristics. First of all, I would like to say you can say the information they have given about lichens. So first of all, I would like to tell you students that lichens are represented here as bioindicators. The first characteristic of lichens is that is their lichens are as bioindicators. Bioindicators. So bioindicators means the lichens, especially Usnea over here, they are the organisms which grow at the place where there is less air pollution. So it means where they are growing, there is less air pollution. So they will indicate the quality of the air. So secondly, they have told us that the photosynthetic cells that are classified in the kingdom Protoctista and the fungal cells that are classified in the kingdom fungi. So it means it shows their symbiotic relationship. Symbiotic relationship. Already told you a type of relationship in which both host and guest they will give benefit to each other. So this highlights the symbiotic relationship between these two types of organisms where they live together and benefit each other. Now outline the characteristic features of the kingdom. So I have written some of the characteristics over here, but I would like to explain more. So the kingdom Protoctista, we will talk about firstly. It is also known as Protista. Also known as Protista. So phylum Protista consists of eukaryotic cells, and you know the eukaryotes have uh, proper nucleus and membrane bound nucleus or other organelles which have cell membrane around themselves, or they belong to the diverse group of organisms. So they can be autotrophic, that is photosynthetic. Already they have told us that is they are photosynthetic. Photosynthetic means they can manufacture their food. So we can say they are autotrophic. Autotrophic or photosynthetic, they can photosynthesize, they can produce their own food. So other characteristic can be the reproduction. They can reproduce sexually or asexually, both methods of reproduction, sexual and asexual both. So they are living primarily aquatic or moist environment. They live primarily at aquatic or moist environment. So they depend. So their habitat is this type of, and their cell walls are made up of habitat, and the examples: algae, amoeba, paramecium, simple molds. For example, algae, amoeba, paramecium. So these are the different types of organisms. So now the next is the kingdom fungi. So again, they are also eukaryotic cells. They also have eukaryotic organisms. That is, the cell possesses a nucleus and the membrane bound organelles. Again, they are only heterotrophic. Opposite to Protoctista, they are heterotrophic. Heterotrophic. It means they cannot photosynthesize or they cannot manufacture their own food. So they depend on other organisms, dead and decaying organisms. So they are living a saprophytic or parasitic life. Saprophytic or parasitic life. Saprophytic, which live at the dead and decaying organisms, and in this way they absorb the nutrients from the soil. And parasitic way of life, the organisms which live at the expense of other organisms. So in this way they are living differently. Some of them are parasitic and some of them are saprophytics, and their cell walls are made up of chitin. Cell walls are chitin. Chitin is another type of material with which the cell walls of these are formed. So they are also living in diverse environment, including soil, decaying matter, and living organisms. It depend on their mode of life. As for example, if they are they are saprophytic, so they will live in the soil, and if they are parasitic mode of life, then they will live on the other organisms. So the examples of them are, for example, mushrooms, which grow at humid places, molds, yeast, and lichens. So the different characteristics. So this is the explanation in detail. Uh, and I would like to read all which I have written. Kingdom Proctista are eukaryotic, which include a wide variety of organisms ranging from unicellular to multicellular organisms, can be heterotrophic or autotrophic, and can produce both sexually or asexually. Question is of 4 marks. We have written almost as, for example, the four different characteristics for the organisms. So kingdom fungi are also eukaryotic and heterotrophic and saprophytic in nature. They reproduce through spores, both sexually and asexually. They found in diverse environment. If we talk about their structure, they have filamentous structure. Filamentous structure. the fungi. We are talking about the fungi filamentous structure consists of hyphae, and group of hyphae is called as mycelium. So it is all about the question number 1A. So next question is B, Xanthoria, lichens that grow in places where there is a high concentration of sulfur dioxide in the air. Opposite to Usnea, Xanthoria is another type of lichens which grow at a place where there is high concentration of sulfur dioxide in the air. It means they are growing at a place where there is high grade air pollution. So, for example, in towns where homes, factories, and vehicles burn fuels. So, these are the places where they grow. They are opposite to the and shows the lichen of the genus Xanthoria. A student planned a method to measure the relationship abundance of these Usnea and Xanthoria on trees along a transect from the town center of at 0 kilometer town center. It means at from the town to unpolluted countryside at 4 kilometer around the countryside, that is about 4 kilometer. So suggest why area from 0 to 4 kilometer. So suggest why measuring the relative abundance of the two types of lichens gives information that is useful for conservation. Well, already in the previous question, I have discussed about that lichens as bioindicators. Bioindicators, here you can see the first type of lichens, they were Usnea, they were growing at a place where there is less pollution. And opposite to Usnea, the Centauria, they grow at a place where it is high air pollution, high grade air pollution. So in this way, I would like to say both of they act as bioindicators. They will indicate the air quality. They will indicate the air quality where they will be grow. So they, I would like to say, if we talk about them, so that Xanthoria can survive in environment with high sulfur dioxide concentrations. So they demonstrate that different lichen species have varying sensitivities in pollutants. So the different lichens, they have lichens. Lichens have differential sensitivity. As one type of can grow in the low concentration of sulfur dioxide, the other grow in the high concentration of sulfur dioxide. So we can say the different lichen species have varying sensitivity to pollutants. So they will have different behavior with the pollutants. So secondly, here we are talking about the transect study. A student is planning a transect study to measure the relative abundance of Usnea and Xanthoria along a gradient from a polluted town center to an unpolluted countryside. This is a common ecological method for studying how environmental factors change over a distance. So transect method can be used and already you have studied in your book. So Xanthoria is a lichen that can grow in a places where there is high concentration of sulfur dioxide in the air. So I would like to say the distribution and abundance of these two lichen species can provide valuable information for conservation effects. So first of all, we can see the data which we collect by the study of these two different types of lichens is number one. Number one type of that data is, I would like to write it, that is this way. The first point can be the pollution monitoring, pollution monitoring. after collecting the data about these two species, we can say we are we can monitor the pollution. So the relative abundance of Usnea and Xanthoria act as a direct indicator of air quality. They will tell us where the air quality is good and where the air quality is not good, or that the air is highly polluted or less polluted. And second point in this regard can be the habitat quality assessment. Habitat quality assessment. So this presence and health of sensitive species, both of they are sensitive species. Both of they are have different ability to or sensitivity against the air pollution. So Usnea indicates the overall health and quality of the ecosystem, and our decline in Usnea populations would signal broader environmental issues beyond just air pollution, such as habitat fragmentation or other forms of disturbance. Means what does it mean? That is, if Usnea concentration or Usnea quantity or percentage is low, it shows that there is high grade population over there. It means we have to think about our environment, because in this way, if there is high air pollution, so it can be a signal for us that there can be the different environmental issues. Number 3 points which we should keep in mind is conservation planning. So the data which obtained from the studies transact can be used to inform conservation strategies. We can conclude, we can suggest if the about the air quality, and due to this air quality data, we can plan to conserve the species which can survive in different environmental conditions. Usnea is a sign of good quality of air, but whereas the Xanthoria indicates the bad quality of air. So in this way, we can monitor the different levels of air pollution, and we can conserve the different species of these organisms which are highly beneficial for us. We can, in this way, we can protect the existing habitats or restore degraded areas. So we can repeated transect studies over time can track changes in lichen populations and monitor the effectiveness of the conservation effects. So this allow and adaptive management and adjustment to strategies as needed. So the data uh, which can be obtained after this study can be used to raise public awareness. The public awareness. Public awareness about air pollution and its impact on biodiversity. Public air pollution and its impact on biodiversity. Biodiversity, variety of different species. If air pollution is there, there will be you can see, there will be the destruction of habitats, there will be the destruction, there will be the changes in the biodiversity. Biodiversity will be reduced, because if there is air pollution, there is lack of biodiversity. If lack of biodiversity is there, it means there will be instability in an ecosystem, which is not and after this instability of the ecosystem, if ecosystem will not exist, so this can lead to increased support for conservation initiatives and policy changes In essence, measuring the relative abundance of Wisnia and Xanthoria provide a cost-effective, relatively simple way to assess air quality, monitor ecosystem health, and guide conservation actions. So the differential sensitivity of these lichens make them powerful tools for understanding and protecting our environment. So we have to work on it. We have to uh, try to conserve. After getting information, we must know with the help of this data, we can conserve these species which are good for us. Um, After this explanation, I would like to read the answer, which I relative abundance of Usnea and Xanthoria act as a direct indicator of air quality. A high abundance of Xanthoria and a low abundance of Usnea near the town center suggests significant air pollution. So the data can be used to raise public awareness about air pollution and its impact on biodiversity. This can lead to increased support for conservation initiatives and policy changes in essence. Measuring the relative abundance of both types of trees provide a cost-effective and simple way to monitor ecosystem health and guide conservation and actions. So these two types of species are very helpful for us because they act as bioindicators, and with the help of them we can better improve our environmental conditions. We can better the place where we are living. We can improve the other resources of life by with the help of these different types of species which are growing over there. Now the next question is part C, and which is although a large biodiversity of lichens can be found in a range of habitats, most people ignore them. Most people ignore them. They are present, but people ignore them. So, um, outlined by forms of life that are usually ignored, such as lichens, should be conserved. Already discussed in previous question that we should not neglect the different species such as lichens, because in this way we are neglecting the biodiversity of the organisms. So they belong to the diverse group of organisms. They are widely distributed and often ignored or overlooked in conservation. We are not working on it. We are not trying to conserve them. So this highlights a common issue in the conservation biology, the tendency to focus on charismatic megafauna while neglecting smaller, less conspicuous organisms. So this question requires us to articulate the ecological, economic, and intrinsic reasons why organisms like lichens deserve conservation attention, even though they may not be immediately appearing as other species. So first of all, we have to uh, keep in mind that is these organisms should be conserved because they are acting as bioindicators. As discussed earlier, lichens are sensitive to environmental changes and serve as valuable indicators of air quality, bioindicators of air quality, and ecosystem health. And secondly, number two, nutrient cycling. Nutrient cycles, lichens, um, they are contribute to nutrient cycling in ecosystem for fixing nitrogen and breaking down rocks, contributing to soil formation. So nutrients recycling is a very important for the growth of the organisms or the growth of the plants. So the food webs, they serve as the food sources. they act as food. They are part of the food webs, we can say that. So they serve as food sources for various animals, including invertebrates and mammals. So in this way, part of the food web. So they are helpful in the food web. Next point can be they are the habitat provision, habitat of many different types of organisms. So lichens provide shelter and nesting materials for insects, mites, and other small organisms. So the another benefit by which for which we should conserve these organisms, they ecosystem engineers. They play a significant role in shaping the physical environment. So they can be called as ecosystem engineers. Biodiversity. They are diverse group of organisms. So I think these are the different points which are enough. Ecosystem stability of the ecosystem. So these are the different points for which these organisms, conservation of these organisms is crucial. Or the conserving organisms like lichens is crucial for maintaining ecosystem health, preserving biodiversity, and recognizing the intrinsic value of all life forms. Ignoring these species can have far-reaching consequences for environment and human well-being. So, there should not be ignorance or negligence about these species or about lichens. So, this is all about the question number 9. Please like, share, and subscribe my channel. Thanks for watching.